I would like to thank everybody for coming to the webinar today. I'm really excited to see all of you here. And I'm also very excited to discuss aged mice, specifically the C57 Black 6J mice with you today, because there's some really important differences compared to younger mice that you need to be aware of. And aged mice might actually be a better fit for your research needs if the human population that you're interested in is also aged. So, before I get into today's talk, I just want to give you a brief introduction to JAX in case this is your first time with us. So, our mission is to discover precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. And we do this by supporting the biomedical community in a couple different ways, from primary research from our faculty and scientific researchers to providing access to services and various mouse models, and through our educational activities, such as the webinar that you're attending today. We also have multiple locations that are marked with white dots on the map here. So in addition to our original location in Bar Harbor, Maine, we also have locations in California and Connecticut, and we've also recently added some locations in China and Japan. Also, all of the countries that you see in a darker shaded blue are locations where we've shipped products and services to. So before we get into today's topic, I am interested on how you're currently using aged B6 mice. So if you're getting them from us or from someone else, maybe you're aging them in your own facility, or if you're just here to learn more. So take a few minutes, well, less than a few minutes, a few seconds, and just go ahead and select which one applies to you, and then don't forget to hit the submit button. So I'll give you a few moments to answer that question. All right, so it looks like the majority of you are aging B6 mice in your own facility. And then we have a few people that have selected all of the other options. So my goal today is to provide some information that's going to be relevant for all of you. Um, and I think especially some of the people that are aging the B6 mice in their own facilities will really benefit from some of the information that I'm going to chat with you today about. So these are my learning goals for today. I'm going to go through 5 major topics. First, starting with key phenotypes, we'll talk about some considerations when designing your studies. I'll introduce you to some resources that are out there that can support your research. And then we'll talk about some specific data around metabolic diseases, cancer, and some neurological changes. So let's start with the key phenotypes of aged mice. And I think the best way to do that is to define some terms first before we move forward. So while lifespan or length of life is a common measure in aging studies, increasingly there's an interest in understanding not only factors that play a role in increasing lifespan, but also factors that contribute to health span or healthy aging. So, health span is the duration of life during which people remain relatively disease free and have a high quality of life. So, aging and senescence, these are all often used interchangeably, although they're technically not the same. Aging refers to the biological change with time across the lifespan, which would also include the developmental milestones of childhood and adolescence, while the term senescence refers to normal progressive impairments in function, such as muscle atrophy, menopause, loss of hearing or vision, and gray hair, for some examples. So let's talk about when mice are old. I know that a lot of people have this question frequently. So age-related disease is not the same as senescence either. I just want to touch on that. Age correlates strongly with a variety of adverse health outcomes, such as cancer and heart disease and arthritis. Um, Mice are also a widely used model for both senescence and age-related disease because of their similarity to humans. So let's talk about the lifespan and how that correlates with human lifespan. So first, the maturational rate of mice doesn't linearly correlate with humans. So they mature much faster than humans in the first few months of their life. It's about 150 times faster, in fact. And during the next 5 months of the mouse's life, it's actually about 45 times faster. And then over these 5 months, the mice reach their mature adult stage, which lasts from about 3 to 6 months of age. And these studies, by the way, were also carried out in C57 black 6J mice with sample sizes of 150 mice per sex. So the mature adult group is used as our reference group or control for any age change. And as this group is past development but not yet affected by senescence. So mature adult mice should be at least 3 months old because although sexual maturity is reached around 35 days of age, rapid growth continues until about 3 months. And that's the equivalent of the human life stage that's about 20 to 30 years old for adults. And then after 6 months of age, the maturational rate of mice is about 25 times faster than humans. So middle age is defined by the fact that senescent changes can be detected in some, but not all biomarkers of aging. A middle-aged group can help determine if an age-related change is progressive or is first expressed only in old age. So for this, mice should be at least 10 months old for inclusion of a middle-aged group. The upper age limit could be around 14 to 15 months, at which point most biomarkers are starting to change. And this phase correlates to humans that range from about 38 to 47 years old. And then in old age, senescent changes can be detected in almost all biomarkers in all animals. So mice in this old group should be at least 18 months old and can extend on to 24 months or even older. So for C57 black 6J mice and many other inbred strains, the upper limit is about 24 months. However, this really depends on the onset of strain-specific diseases, which can affect biomarkers and produce misleading results. So with advanced age, necropsies and pathological assessment become really essential for accurate interpretation of the data. Many of the histological lesions associated with aging are also very apparent by about 24 months of age. And so mice to the right of this old group might be considered very old, and survivorship is going to start to drop off dramatically. So now that we've discussed age and what is considered old, let's talk about the associated phenotypes. On our strain data sheet for aged mice, there's a section titled Appearance and Care where all of this information can be found. The first thing I'd like to note is that the coat can become thin or gray. And additionally, there can be areas of alopecia or hair loss, which can be due to over grooming by the animal itself or another mouse in the cage. Hair loss from being overgroomed by another mouse is called barbering. And although this is a normal mouse social behavior, it can become extensive and quite striking in appearance. Barbering can also result in the removal of whiskers from the muzzle, like you can see there in figure B. And these are all normal characteristics of aging and are expected in this colony. Um, Age cohorts of mice might be shipped with hair loss or thinning hair and whiskers, and it's not related to aging. However, we don't ship any mice that have inflammation or dermatitis. Now, there was also a study conducted independently at the University of Washington using C57 black 6J mice, and they described the most um, common clinical signs in the aged B6 colony that was housed in their vivarium, which included both males and females between 16 and 36 months of age. And these mice showed four common clinical presentations. First was rectal prolapse, as seen in Figure B, alopecia and dermatitis, ocular lesions, as seen in Figure C, and palpable masses. Now, I'll only present a summary of the study findings, and I would highly recommend and encourage anyone who's interested or plans to work with aged mice to read this original reference for an extended discussion of the various differences that you might see in these aged mice. So this here is a survey of all of the neoplastic lesions that were seen in their study. So let me orient you here first. So in this study, there were over 700 mice that were used. This data presented here is the percentage of mice that displayed a neoplastic lesion. So in this case, 72 mice presented with neoplastic lesions. And for example, 67% had hematopoietic neoplasia. So veterinary staff might be unaccustomed to working with aged mice and might benefit from some of this orientation to what kinds of abnormalities to expect. And as discussed, advanced planning on how to handle these issues. So be sure to manage the expectations of the veterinarian facility staff. Make sure that they're aware of some of these abnormalities, as your mice might get marked as sick several times. So the stakeholders of this study and animal facility really need to discuss the fact that not all lesions represent sources of significant distress and are not necessarily life-threatening. Um, So here is also a table of the non-neoplastic disease among the age C57 Black 6J colony at the University of Washington. And like I said, these percentages here are reference of the mice with non-neoplastic diagnosis. And of note, these lesions observed in the age colony at the Jackson Laboratory differ from those at the University of Washington. And this is likely because the age group at JAX is relatively younger on average, and the UW cohort was also maintained at a different health status. In the aged colony at the Jackson Laboratory, ulcerative dermatitis is sometimes observed, as well as a few other diseases. These differences accentuate the point that different phenotypes and pathology can be expected to emerge as a difference of age, sex, bias, and cohort. So now that we know the commonly associated phenotypes in the age C57 Black 6J mice, let's discuss how we might study those phenotypes in aged mice. So the first thing to consider when preparing for your study is how are you going to identify your mice over the course of your study? And there's several different methods out there for identification, and many have different pros and cons. And so it's important to consider how and which method you're going to use. So for example, if you're going to study a mouse over the course of its lifespan, you might want to pay attention to your ear punches because over time they could become torn, and you might not be able to distinguish two mice from each other. You might also want to consider using ear tags or microchips. You could also even use tattoos. Um, really think about some of the pros and cons and how they will fit with your study before deciding on a method. The next thing to consider are what assays you're going to use in your study and how you're going to select which assays to use. So I have listed here several different assays that have been used to study aged mice. And for example, you might want to consider um, the electrocardiogram or the echocardiogram. Those require anesthesia. And so that might require some additional planning um, just because there is more risk at putting aged mice under anesthesia. So that's something to consider, which brings me to my next point. Like I mentioned on the last slide, some of these assays that you might want to run could be invasive, like the electrocardiogram. And so you're going to need to determine the order of your assays. And one very important consideration is the invasiveness of these assays. Do you want to run the assays from least invasive to most invasive? Is it important for your research question that you balance your groups so that the order of the assays are accounted for? And might running one assay first impact the outcome of another assay? So these are some of the questions that you'll want to consider as you plan the order of your experiments for your aged mice. And finally, it's also really important to validate the assays in your cohort. So you need to keep in mind to control for things like age, sex, and environmental conditions. So let me give you an example of this. Because the environment can exert such a strong influence on phenotype, it's really critical to include the proper controls. The ideal controls are age and sex match litter and colony mates. So shown here is data you've probably seen before. It's the survival curve for the C57 Black 6J mice that's been adapted from data contained in the mouse phenome database. Showing on the x-axis is time, age in weeks, and the y-axis is the percent survival of the cohort. Overlaid in the dots are the survival data taken from the National Institute of Aging's website based on a 1999 reference in which in which a different substrain of C57 Black 6 was studied. The strain was the C57 Black 6N NIA, and the time at which 75%, 50%, 25%, and 10% of the population has survived is shown with green and red dots for males and females, respectively. These ages are derived from large populations, so small data in small groups of animals will vary. So in this study, the mice are a different substrain of C57 Black 6. It's the N substrain. And you'll note the different lifespan than J. The data that's presented here is to emphasize how essential it is to include the appropriate controls in your experiments. The different substrains of C57 Black 6 are not the same strain. And additionally, housing can exert a strong influence on the phenotype over time. So it's recommended that investigators house control groups in the same facilities as the experimental group, so they're subject to the same environmental factors. And of note, the NIA now distributes mice from JAX, and you'll note that the nomenclature C57 Black 6J NIA or C57 Black 6JN. The J after the mouse strain indicates that the foundation stock came from the Jackson laboratory. And if the suffix says JN or JNIA, this indicates Jackson stock but bred at the NIA colony. And lastly, also of note, the NIA will not ship animals past 25% survival age, as very old mice are frail and become sensitive to the stress of shipping. And the oldest that we ship mice or tissues is 90 weeks of age. So let's move on now and talk about some of the resources that are available to support your aging research. The first resource that I would like to share with you is the Nathan Schock Centers of Excellence in the Basic Biology of Aging. And JAX is one of those centers. And we have many experts conducting aging research at JAX. The link at the bottom of your screen will take you to our web resource where we have lots of information that might be useful to you. And here you can find data on aged mice, some tools for analysis, and related publications. So I highly recommend taking some time to look through these resources more in depth. Some of the studies that have come out of the Nathan Schock Aging Center at JAX are listed here and can also be found in the mouse phenome database. The graphs on the right show the survival fraction of many of the commonly inbred mouse models available. And you can see, for example, in the red box, the C57 black 6J male mouse survival fraction in the lime green line. We also have collected quite a bit of data on our aged C57 black 6J colony here at JAX. We have mice ranging from 25 to 90 weeks of age ready to go on studies. We also can combine these mice with additional breeding services and aging services. We also maintain these, or we can also maintain these mice on special diets at your request. We can also send you tissues from aged mice so that you don't have to do the tissue collection yourself. And of course, we can always run in vivo studies with our aged mice to help expedite your research goals. But as I said, we have quite a bit of phenotype information available on the strain datasheet. We currently have data available for mice at 26, 52, and 70 weeks of age, looking at things like organ weight, hematology, body composition, serum clinical chemistry, and flow cytometry. So now let's transition and look at some data for our age-related immunological deficiencies, specifically looking at metabolic disease and cancer. So it's important to know your mouse model before you begin your experiments. And one example I like to use is the body composition changes seen in the C57 black 6J mice as they age. Here you can see the percentage of body fat for males and females. And what I'd like to point out is that between 6 and 12 months of age, both groups gain body fat. However, between 12 and 20 months of age, body fat percentage decreases, which might be important for you to know if you are planning your studies around body composition. And in addition, there are also age-associated changes in the clearance of glucose, as shown here. The young C57 black 6J mice around 4 to 5 months of age, while the aged mice are 24 to 25 months of age. So if you're looking at a metabolic phenotype, it's important to know that they can change significantly over time, which may affect when you perform your experiments on a cohort of mice. So within our facilities, we've performed glucose tolerance tests on both male and female mice and confirmed this finding. As you can see here in the graphs, glucose tolerance decreases with age. The data that were collected at several time points and show progressive worsening of glucose tolerance until at about 18 months of age when the mice are just not able to recover from a glucose challenge. Body weight also continues to increase with age, but also increases in variability with age. So in our 90-week-old male mice, we expect that we'll see some mice that are 30 grams, and we might see some mice as much as 60 grams. So this is also really important to take into account if body weight will be a factor in your study. We and the NIA are unfortunately unable to select mice for a shipment based on age. So you're going to want to make sure to account for that in your sample sizes and size your groups appropriately. One hot topic in the metabolism field is how to regulate a lot of negative metabolic impacts of aging. And a common experimental design is used by using caloric restriction. So caloric restriction has been shown to reduce obesity, but also increases lifespan and health span in rodent and non-human primate studies. There is an increase in metabolic flexibility that leads to a decrease in inflammation, which we'll talk about a little bit more in the next slides. And also there is a decrease in age-related disease thought to be mediated, at least in part, by an increase in DNA repair. And all of this accumulates in an increase in lifespan. So we already have a ton of published data showing that caloric restriction can lead to an increase in health and lifespan. So researchers are now taking this one step further and thinking about which component of the diet is likely to have this impact. This particular study looked at diets that contained the same number of calories but were made up of different nutrients. And after 12 months of age, mice were placed on either a control diet, 65% kilocalories from carbs, a low carb diet, or a ketogenic diet that contains almost no carbohydrates. And you can see that the median lifespan is significantly increased from the control to a low carb diet. And this is further increased by the ketogenic diet. An interesting finding here is that the fat mass, which actually increases on in the low carb fed group, is similar to the control group when fed the ketogenic diet. Looking more functionally as well, they observed skeletal muscle strength at older ages and found that there's a significant increase in grip strength and hanging time with the keto diet group compared to the other two diets. So the main takeaway point from this study here is that it is likely not just the number of calories that matters. The specific composition of the food may also play an important role in aging phenotypes. So now caloric restriction is something that very few people actually want to practice. I know I certainly don't. However, gaining insight into the pathways that change during caloric restriction provides potential pathways that can be targeted to result in the same effect. So, one possibility is hormone supplementation. And one particular study determined that by treating aging C57 black 6J mice with 17 alpha estradiol, they were able to modulate the amount of both visceral and subcutaneous fat that these mice had. And there is a significant loss in total mass, and most of it is due to the fat mass and not lean mass. Importantly, there is a large concern whenever you're using a hormone therapy that there can be other negative effects. And this group also characterized that there were no cardiac dysfunction or feminization of males with this particular treatment. Um, so what they noted was that there was also a decrease in many pro-inflammatory markers, such as TNF-A alpha and a few others. They also noted this at the mRNA level as well as in the plasma. So, on the topic of inflammatory markers, in your adipose tissue, you always have a few macrophages that are present. But as the amount of adipose tissue increases, this recruits more macrophages into the tissue. This increased recruitment of macrophages then increases the adipokine secretion of pro inflammatory cytokines, which in turn leads to adipocyte death. And this further recruits more macrophages, which triggers a whole cascade of events that leads to a pro inflammatory state. This is shown with histology here. So, in a lean and obese mice, you can see that as obesity increases, there is the same recruitment of macrophages. These macrophages are pointed out by the arrows in the images. In the big B6AY strain, it's a moderately obese strain, but looking at the DIO mice or the diet-induced obese mice, or the mice that are the OB, lacting leptin, there are high levels of macrophages being recruited into the tissue. Now I'm going to move on a little bit and talk more about the immune system and immune senescence and show one of the hallmarks of aging in people, and that's thymic involution. And this is where the thymus, where T cells are matured, begins to decrease in size. And as you age, There is a natural decline in the naive T cell output and shrinking of peripheral T cell pools. And as you age, there is a decrease in immunity. And when you are able to clear an infection, it tends to take quite a bit longer. So, this is one reason a lot of people are studying the thymus, specifically in aged mice. So, mice, similar to humans, have this loss of T cell repertoire as they age. And as you can see here, the majority of T cells in this young mouse are naive T cells in the green box, and they can react to new infections and stimuli. While there's still a smaller population of effector and memory T cells in the red box. But as you can see here in the older mouse, at 18 months of age, we can see that there's a decline in the number of naive T cells, again in the green box, and an increase in the effector and memory T cells in the red box. So as the mice get older, just like people, we see this decline in naive T cell populations. So they're not going to be able to respond as efficiently to new challenges. And as a quick aside, we're excited to share that these are data from our own age colony, and we do indeed see these changes in the CD4 and CD8 T cell populations in the aged mice available here at JAX. So let me orient you quickly to this graph. In the far right, the first column of data in the naive group are the young male mice in blue and female mice in red, aged to 2 months. And in the left side of the column are the old male and female mice aged 18 months, again in red and blue. Comparing the two columns, we can see that the naive CD4 T cell population is dramatically decreased in the aged population, and the effector memory cells have also increased. Now, I'd like to jump back to those original investigators who saw this phenomenon. When they looked at expression of genes in the thymus of young and old mice, they saw a decrease in the FGF21 expression as mice age. They then overexpressed this gene in the thymus, and what they found is that overexpression of this gene increased significantly the naive T cell population, as well as decreased the effector memory T cell population in aged mice. They were also able to show that this overexpression actually increased early thymocyte progenitor cells, which allows for the possibility of more naive T cell production. And these numbers are incredibly low, but this is actually an increase and may be able to have a significant effect. One of the reasons this is becoming so popular to study is because one of the possible therapies being looked at for cancer is actually immunotherapy, getting your own immune system to attack the tumors. However, all of these compounds are T cell mediated processes. Therefore, having a T cell population to work with is critical in having these potential drug therapies work. There are a couple of examples in some of the common drugs that are available to human patients. Yervoy is this anti-CTLA4 drug, and Keytruda, which is an anti-PD1 drug. So basically, both of these drugs block T cell pathways that are normally activated in normal cancer progression. There are many other drugs out there, either used alone or in combination with others to generate the best effects. However, the vast majority of these are targeting T cells. So it's critical that T cells are present for you to have a cancer immunotherapy that can actually work. However, cancer itself can also be considered a disease of aging. So people that are 65 years or older account for over 60% of the new cancer diagnoses. But it's important to note that immunotherapy can induce systemic toxicity that was not observed in animal studies. However, when looking through the literature, that often these immunotherapy studies are often performed in relatively young mice, not old mice. So how might this affect the results of a study? I'm going to show you how critical that it is in this example. So this group previously had shown that the anti-CD40 monoclonal antibody in combination with an IL-2 that was able to induce regression in a metastatic renal tumor in 2 to 3 month old C57 black 6J mice. They came back and tested the same therapy in a 12-month-old mouse and had the same or that had the same tumor. And what they found was that 2 days post-treatment, there was a 100% mortality rate in all of the older mice. They went and found that the toxicity didn't even require the cancer to be present. Just the treatment alone could kill the mice. So they performed a time analysis and found that when they looked at 6-month-old mice, the data is not shown here, but none of the mice died. But at 9 months of age, about 50% of the mice died with the treatment. So basically, they saw that toxicity starts somewhere between 6 and 9 months of age in these mice. And they also saw that there was a significant increase in pro-inflammatory cytokines in these older mice, in the mice that received the therapy versus those that did not. So here, they also showed the toxicity effect in the liver, looking at ALT values, showing toxicity is due to liver toxicity. To investigate what pathway was responsible for causing the inflammatory response, they used interferon gamma receptor knockout mice and TNF knockout mice and repeated their experiments. In the interferon gamma knockout group, they saw 50% mortality compared to the wild type mice. And then when they looked at the TNF knockout mice, they saw no toxicity. This is compared to the 100% toxicity in wild type mice. And so this indicated that the lethality is predominantly dependent on TNF levels, which are shown here. So to tie this all together, we know that there are other things that can cause inflammation, such as adiposity. So these investigators followed up with a final study that looked at obese mice to determine if the drug will cause lethality as well. And they looked at two models, diet-induced obese mice, as well as mice homozygous for the OB mutation, which leads to obesity. And they found that the OB mice had 100% mortality, similar to the old wild type mouse model. And the DIO mice had a partial toxicity. So when they looked at TNF expression, they saw that young wild type mice have low levels. But young OB mice have significantly higher levels, and so do the aged mice. And the DIO mice were in between the two measurements. So as the mice get older, they have this increased inflammatory state that can cause issues with toxicity of drugs. Um, but obese mice and even young obese mice can show similar increases in inflammation. So now that we've covered the immunological responses in aged mice, let's wrap up talking about some of the neurological changes that are seen in aged mice. So as we just discussed, this is really a cycle where the increase in adipose tissue leads to secretion of adipokines and inflammatory factors such as IL-6 and TNF. Um, and these go into the circulation, and this low-grade activation can cause systemic inflammation, which can also activate neuronal pathways that can lead to downstream behavioral alterations and neuropsychiatric symptoms. So let's look at some of these neurological changes. This research group showed that there were no changes in brain volume as mice age. But as in humans, gray matter rich regions decline with age, and this was also seen in the mice, specifically looking in areas like the thalamus and cerebral cortex. The ventricles and white matter actually increase in aged humans, and this is also seen in mice, specifically in areas like the corpus callosum and the fornix system. So many studies have reported behavioral dysfunctions due to aging in mice. And this study here compares many behavioral tests and many different age groups and found significant behavioral differences between age groups in all of the behavioral tests that they looked at, including light dark transition, open field, elevated plus maze, rotarod, social interaction, um, and Barnes maze, as well as many others. And so this type of work really demonstrates the importance of the question, when do these neurological changes occur? And is there a particular window where a therapeutic might be most effective? So here, impaired spatial learning and memory is shown in older C57 black 6J mice. Um, and we can see that in these graphs here. So here's another example in older mice to see if you can improve memory in aged mice. So here they were looking at behavioral separation ability. So if you have mice in a cage with an object, and then you move that object a distance that is far away, in this case 35 centimeters, mice are able to know that the object has moved and they will go to investigate the object to see what's happening. But if you move an object a shorter distance, such as 10 centimeters, the mice won't spend as much time investigating the object because it's not as big of a change to them in their environment. But if you move the object a medium distance, such as 20 centimeters, the younger mice will pick up on the fact that the object is moved, or that the object has moved, and will spend longer investigating the object. But older mice won't. So the researchers here treated the mice with D-serine to see if this would help ameliorate the cognitive decline in older mice. So at 35 centimeters, all of the mice noticed the object had moved. But at 20 centimeters, the D-serine treated mice actually had some cognitive rescue, and those treated mice were spending more time investigating the object. So it's worth pointing out here that these mice were treated once they had already aged, which is translationally relevant because in the human population, we wouldn't necessarily begin a therapy until we noticed that there was a cognitive deficit. All right, so that brings me to the end of our presentation. I hope that you learned about some of the key phenotypes that are present in aged mice. And we'll remember to work with your veterinarian and facility staff to make them aware of those phenotypes. And we covered some considerations for developing your studies using aged mice, making sure that you're looking at the appropriate time windows um, and the appropriate assays. I also covered some of the resources that are available for your aging research. And then we talked about some of the data that is associated with aged mice related to metabolic diseases, cancer, and neurological changes. So with that, I would just like to remind you that Jax is here to help you along your research journey at whatever stage that you're at. If you are just at the model access stage where you're interested in mouse models, we're there to help you with that. We can also help you characterize your mouse model, and we can also help you with your preclinical solutions.